Mani is going to talk about a method to discover non-linear causal relationships among the genes. Yeah, Mani, to you.
Like Nirav was saying, I'm going to talk about the nonlinear causal discovery method, and it's a great pleasure and honor to be here. So there are all these dense kind of meeting schedule we have at IMSC, thanks to Arijit and also co-organizer Maya for this wonderful opportunity to be able to share some of my work. Okay, and this one is about identifying relations between genes, causal relations between genes from observational data. So these are, you don't have access to experiments where you can actually knock out a gene or knock down a gene. Then the problem is more straightforward. You knock out a gene and see which other genes are being perturbed, and then you can establish relations. But in human settings and different other settings where it is not ethical or not feasible to knock out genes or knock down genes or overexpose genes, do any kind of perturbations, what can we do to infer gene-gene relations from observational data? Okay. And I would like to start with acknowledgements first in the manner that Arijit was also acknowledging people. So like I said, it's really nice to have these frequent meetings here where we can exchange our ideas and then meet with other leading computational biology researchers across the country. And this talk particularly is going to be a work that was led by Arvind Ishwar and also joint work with Neeraj Parmar. This is on, like I said, on identifying nonlinear relations between genes. But I would also like to give a brief context about our group, Bioinformatics and Integrative Data Science Group, BIRDS group we call it, before delving into this particular causal discovery problem. So this is the recent calendar, 2024 calendar for IIT Madras. Those from IIT Madras would know. So we basically different birds in the campus were featured in this calendar. So they are all of different colors, different feathers, different traits. But what I would like to say is they call the campus the home. So similarly, the members in our group have very diverse interests, but we are shared by a common research theme of using the power of computation to tease apart the gene or molecular interactions underlying life. Okay, and that is really the theme of our group. And we work on broadly two areas. So one is multi-tissue or multi-organ genomics. We want to understand the communication between different tissues and how does it happen at the molecular level. What is the molecular basis of gene-gene? What is the molecular basis of tissue-tissue or organ-organ communication? For that, we have developed different graph algorithms and probabilistic models for understanding and modeling these communications. And the other part is about disease genomics. So one of the posters, for instance, by Nilesh, who is in the audience and outside, is on disease-disease interaction. So how do we analyze genomic data that is coming from different cohorts of combinations of diseases and also host pathogen interactions? Again, some of my group members are in the audience who work on these areas, and you can interact with them during the breaks. But today's focus is on causal discovery, and then one of the main language that we use from computer science to understand biological system is the language of networks, where you have a set of nodes, in our case genes, present in either one tissue or different tissues, and then relations between them, which are the edges of the network. So how do we infer this network in the first place, and then how do you analyze these networks and to derive biological insights, and subsequently collaborate with experimentalists on the hypothesis that we have generated to validate them, right? So that is the broad theme. And the very first step is how do we get these gene networks in the first place? There are various approaches. You can use standard kind of literature-based approaches where you have a database of protein-protein and protein-DNA interactions, transcription factor target gene interactions, or protein-protein interactions. Or you could look at correlation or co-expression-based approaches where you say that if two genes have co-varying patterns of expression across a set of measurements, then you assume that they have some form of functional relationship. But we would love to get deeper and then identify which of these correlations that you observe between two genes are actually causal. Okay, if two genes A and B are correlated, is A causal for B, or is B the regulator for A, or is there a third factor, a confounding factor, which is just simply driving both A and B, and then they have these spurious correlations because of this underlying confounding factor. Okay, and this is not of particular interest to only understanding gene-gene relationships, but it has a broader interest in terms of causality. Trying to differentiate causality from associations is a holy grail, right? In many sciences, including economics, social sciences, and also biological science. But how do we infer causation and separate it from correlation from purely observational data? Like I said before, you just have a random set of individuals, let's say from a population you have sampled, and you have measured the gene expressions or additional variables in that population. Then how do you infer causal relations? Let's say that each dot here represents a particular individual. Okay. Yeah. So each dot here represents an individual that you have sampled from a population randomly. And you observe that two variables, in this particular case, an expression trait A and B, are correlated. Like I said, the main issue is that this could be either gene A is causal for B or gene B is causal for A, or it could be that there is an underlying confounding factor that is driving both. And then because of that, these two look correlated. Okay, the underlying factor could be other genetic factors, or could be gender. Maybe these two genes are differentially regulated in males versus females. Because of that, you may see a correlation. But within each of the group, within the males or within the females, once you condition on each of the group, then there is no relation between the two. Right? So how do you separate out those scenarios? And this is just a standard, an illustration of the standard thing. Correlation does not imply causation. And one of the things is this, a randomized control trial. If you can do perturbations, you can do randomized control trials. And then the way that it modifies this is according to Pearl, who formulated different foundations of how to represent causality and causal inference tests. So you just take a particular gene A and then you set it to a particular variable. If you can do perturbations or interventions, right, knockouts or knockdowns or overexpression. So you just affect only this gene and set it to a value A. It could be a low value A, it could be little, it could be 0 or a very high value, right? And then you see what is the effect on B, and then you set it in a way that then this relation between C and A is broken. Because regardless of whatever genes or factors are controlling A, you just force A to be a particular value, right? This is the idea used in randomized control trials, where you just randomly distribute patients into two groups and give one a drug, another a placebo, right? So a similar idea can be applied to perturb genes and identify these causal factors. But of course, we don't have access to these trials in a human setting where it may not be feasible or ethical. And so how do we fix it in the presence of observational data, right? And for that, actually, genetics gives a beautiful solution. So in the form of Mendelian randomization, some of you may be already aware of it. Mendelian randomization is like randomized control trials performed by nature. So when we are born, when the embryo is formed, right? So we have like each of the egg and the sperm cell that contribute to the embryo are already randomized in a sense. So the different genetic factors are we are getting randomly from either our father or our mother, right? So L is the genetic factor that is correlated to these genes. And L is already randomized, and because of this randomization, it inherits some of the nice properties of these randomized control trials, and you can perform your causal inference or causal discovery using a third variable called L, okay, the instrument variable or the genetic factor in our particular case, okay. Without this third variable, you will never be able to distinguish between whether or any other extra information, whether A is causal or B is causal, or whether there is a confounding factor. But with this third variable, L, you can use it as some kind of a causal anchor. We call it as an instrument variable to figure out whether there is a causal relation or not. Okay. And there are certain assumptions behind this concept of Mendelian randomization. As with causality, there are always assumptions. It's a very heavily loaded field. There is a lot of philosophical books written about causality, like entire books written. We are going to go with a simple operational definition that if one gene is affected in this do calculus way, that you only affect that gene and no other gene, does the other gene get affected? Okay, so that is the definition we are going to go with. And in order to identify that, then you need an instrument variable like L, which is correlated with both this genes A and B, but it should not have any independent effect through some other pathways. Okay, it has to always, the effect of L on B has to go through A, right? So that is the assumption that Mendelian randomization takes, and then it's also not correlated with the confounding factor. So if under these assumptions and the randomization assumption of L, you have a series of statistical tests that you can perform to figure out if A is causal for B or otherwise. Okay. And people have, researchers have done a lot of different extensions of this basic idea to infer gene regulatory networks. And most of them focus on linear relations between genes like trigger, causal, finder, and there's MRPC methods. And there are a few of them recently that are coming up with nonlinear, but they don't handle general nonlinear relations. So PolyMR handles only polynomial regression-based relations, for instance. And so this is where our method fits in, or our goal comes in. Okay, so our goal is to develop a pairwise causal discovery method that can detect nonlinear relations between genes. Linear relations are also included as part of the discovery, so it should discover both linear as well as nonlinear relations between genes. And the specific problem statement is that you're given these three variables. L is a genetic factor like a single nucleotide polymorphism or a SNP that is correlated to two genes A and B. So we also call this as EQTL. So those who work in the area of expression quantitative trait loci will know that if L is a variable, so for people who are not familiar with these terms, you can just think of L as simply a binary variable 0 or 1, right? It's a discrete random variable. And it changes across the population depending on what allele is present in that particular SNP across the individuals. Okay, so L is a discrete variable. A and B are continuous variables, measures of gene expression across the same population of individuals. So LAB is measured in the same individual across a population of individuals. Okay, so that is the data that you have. And you want a nonlinear causal discovery method that will try to distinguish these three models, whether B is causal for A or A is causal for B, or whether there is no relation between them and they appear correlated because of this underlying factor L that is driving A and B independently. Okay, so that is the overall model. So we call it a trio because there are three variables here. A and B is our primary variable of interest. L is just the additional variable that will help us distinguish between the three models. Okay. I'll pause here and then see if there are any quick clarification questions. So, yes. Yes. So you need both matched gene expression and genetic data from the same population of individuals. They are all from the same population of individuals. We need genetic and gene expression data from the same population of individuals.
Okay, and if you don't have, then there's no possibility of you having L as an anchoring parameter.
That's right. In our method, we cannot have. There are some other methods in Mendelian randomization called two-sample MR, in which you don't need all of them to be in the same population. But for our method, we need all three to be in the same population. Yeah.
But, it might happen that, let's say, B is affected by not just A, but by other things in the system.
Yes.
Which you may not know. I mean, you may know the value of L, A, and B.
Yes.
But you don't know the values of the other variables which are affecting B.
No, very good point. That's what I'm going to come next here. So there is a theorem called causal equivalence theorem, where under certain assumptions, right? So only these assumptions are required. So the L has to be randomized, which we assume happens because of these random crossovers and recombinations that happen during meiosis to produce the sperm and egg cell, right? So this L is randomized, meaning it takes the value 0, 1 in terms of haploids or 0, 1, 2 in terms of diploid organisms like humans. And if L is associated with A, meaning correlated with A, if L is associated with B, so they all have to be EQ12 relations for us. There has to be correlation to begin with to test for causality, right? So, and there is this key third test, L is independent of B given A, right? So once you condition on A, once you stratify your samples based on a particular bin of A, then the association between L and B should be lost. So what does that mean in this particular scenario? So let's look at this model. L should be associated with A, L should be associated with B, and then once you condition on A, there is no other path where L and B are correlated. If those conditions are met and L is randomized, right, then you don't have to worry about any other variables in the system, okay? So the theorem basically says that the causal relationships L to B, A to B exist, and there are no hidden variables causal for both A and B, so you don't have to worry about confounders, okay? So this is, you may wonder, this is a very restrictive setting. Are real A and B, will they satisfy this conditional independence, which seems a very restrictive test, but that is kind of the a limitation of any method in this framework. Like all the methods that I listed in the previous slide, all of them share this thing that they will be able to identify causal relations, but they won't be able to identify all causal relations. So there will be a lot of false negatives, but then whatever it identifies, we are confident that they are true causal relations. Okay, so there is a trade-off here. Okay, but that is a good point. But we don't have to worry about any other variables in the system if L is randomized and all these conditions are met. And all these methods, including our method, work under this assumption. And the main distinction between the earlier methods and our method is that all the earlier methods, or most of them, are based on assuming linear relationships, and they implement these statistical tests using linear relations. We have extended it to the setting of nonlinear relations. Okay, that is our main contribution here. So this 3 tests can be extended into 4 tests which have better empirical performance. I'm not going to go into it into particular detail, but I just briefly mention it because this causal inference test CIT from an earlier method is our baseline method that implements the statistical test under the linear setting and identifies linear relations. Okay. And let's just briefly look at why is this able to tell us, this conditional independence test, why is it able to give us the clue about the 3 different models, right? So if you condition on this confounding variable, G and T are independent if you have this independent model, but if you have these other dependent models, right? So if you have the dependent models, like, I'm just using the terms G and T for A and B, apologies for switching that, but G is causal for T or T is causal for G. If you condition on G, the relation between L and T is lost here, right? If you condition on T, the relation between L and G is lost here, but here, conditioning on L, the relationship between G and T is lost, but not based on G or T, okay? Using that, these 3 tests, so is G correlated to T? In all these cases, the 2 genes are correlated, okay? That is the standard correlation doesn't imply causation issue that we have. But once you do these conditional independence tests, conditioning on G and conditioning on T, then you will be able to distinguish between these 3 models, okay? I know, but for a short talk, this may be kind of, if you don't know Mendelian randomization to begin with, this may be a little bit fast, but I can address questions later on. But what I wanted to convey here is that the conditional independence test is the key here, okay? If you have a model like this, if you condition on G, then you're blocking all the information that L carries through to T, and so L and T association is lost. But on the other hand, if you condition on G here, L and T association remains, okay? And vice versa if you condition on T, okay? So what we have is, I'll just briefly say how we implemented. I won't get into the details. I will just talk about just one of the tests. Again, there are 4 different tests that we implement in the nonlinear setting. Only one of the tests, which is the conditional independence test, is the most important test. Okay, so I'll just go into that test alone. And this is kind of an overall summary of all the 4 tests, right? So we want to distinguish between these 3 models. A is causal for B, B is causal for A, or A and B are independent, okay? And then there are these different association tests. The first 3 tests is L associated with B, is L associated with A, is A associated with B. These are all correlation tests. These are all kind of standard because you need to make sure that L, A and B are correlated to each other before you test for causality, right? So the first 3 tests are standard. They are implemented in a linear setting there, and then we were able to use nonlinear regression. NLR stands for nonlinear regression. So these are standard neural network or kernel ridge regression or other kind of machine learning models that will model nonlinear relations between 2 variables, okay? And then NLL is a likelihood ratio, negative log likelihood ratio that we use in a permutation setting to get a p-value for each of these first 3 tests, okay? But like I said, the 4th test is our main contribution. So we are using nonlinear regression and something called conditional feature importance. So this is like traditional machine learning models, you have a feature importance. For every feature that you are using to predict the outcome, you have an importance for that feature. Now we want to do conditional feature importance, that if you condition on A here, right, is L important for predicting B, right? If L is important, then you know that this conditional independence test fails. If L is not important, then this test passes. Okay, so that is the key idea. So you use, I want to check if conditioned on A, if L and B are independent, right? To do that, I'll build a regression model, a nonlinear regression model, where I use L and A as input features to predict B, right? Remember that L, A, and B are measured from an individual across a population of individuals, right? So you just measure L and A across a set of individuals and use that to predict the B, that is the B hat. And then once you're providing this A, conditioned on A, is L really informative, or does it provide any extra information on top of A to predict B, or is it just redundant if A is already provided? Okay. So that is the independence test, and then we use this conditional feature importance score to implement this test. Okay. So if there are questions about the technical details, I can answer towards the end. Okay. But what I wanted to show you is some kind of a figure on what this conditional independence does, like a plot. Okay. I'm going to skip the next set of slides. Yeah. So this is so this usually these plots are kind of more informative than the equations in my view in a short talk. So let's just look at this two variables A and B. Okay, they have a nonlinear relation. In this particular case, they have a sign relation. Okay, and then you have these two genotype settings. Right, we have a SNP that is either the minor allele or the major allele. Right, so 0 and 1. So you have these two colors. Right. That represents all the individuals for which at this SNP you have one allele or the other allele. Okay. So now you want to learn from this plot whether A is causal for B or B is causal for A, or A and B are independent of each other. Okay. So in this particular case, we have generated it in the setting where L drives A and A drives B. Okay. A is causal for B. So now what we do is that we use this information of A to predict B. And so, if you think about it, if any simple nonlinear regression model will just fit a sine curve through it, right? If you give it enough number of parameters, it will be able to predict, you put a line through the data cloud of points here and then infer the relation between A and B, right? Now you ask, if you only look at L equal to 1 points or L equal to 0 points, will you infer two different curves or the same curve, right? So, that is the key principle behind conditional independence test. If you get two different curves, if you fit the model for L equal to 1 and L equal to 0 separately, then L carries extra information on top of A, right? If you have the same curve for both of the strata, then you know that L doesn't provide any extra information, and so L is conditionally independent of A given B. Of B given A, right? So that is the idea, and that is being and that needs to be tested only in this overlap region. So that is our contribution. So before we were looking at across the but then these machine learning models are built only in the area where there is enough training data points. Okay, if you go too much to the left or too much to the right, then the fitted curves are varying wildly, and so you don't want to compute your feature importance in those regions. So you want to only compute the feature importance in this gray bar where both of the L equal to 0 and L equal to 1 values overlap. Okay, and that is provided by the second term, this min term. And then the first term basically tells you is there a difference between the two curves that you fit on this data. Okay. So given that kind of introduction to the background of the causal equivalence theorem and an implementation of the causal equivalence theorem under the nonlinear setting, that is what we call as the NLCD method. Then we applied it to different types of simulation data sets. One where the distribution of a gene conditioned on the genotype L equal to 0 or L equal to 1. Are of equal variance and unequal variances. In real world data, they may be of unequal variances, and finally to real world human data in this particular case, but we have also applied it to yeast data. Okay. Let's look at simulated data sets where you have equal variance, meaning that if you look at the width of this of these two data points, right, yellow and pink here, so they are of the same width, right? So this A given L equal to 0 is over here, A given L equal to 1 is over here, and then there are different types of relation between the variables. Okay, so if there is a causal relation, it could be a parabolic relation that is shown in the middle, it could be a linear relation. This is the setting where earlier methods like CIT were developed, right? So they will work quite well in this setting. And even for parabola, you can see that depending on the nature of the curve, it can be approximated by a linear relation. So even parabolic relations can be inferred by linear models. But then these sine and sawtooth are scenarios where they are favorable for our methods, or where our methods are targeted to find these kinds of relations, right? And CIT or other baseline methods are not explicitly designed for this, okay? An independent model is this model where there is a correlation between A and B overall, but if you stratify within L equal to 0 or L equal to 1, you get this big blob. Okay, there is no real relation if you condition on L. Skip the parameters we used and then go directly to the precision recall curves. So the way to understand this is that we generated different trios according to these different relations, linear, sine, sawtooth, and so on. So this first column is linear performance, the second column is sine performance, and the third is sawtooth performance, okay? So we have different versions of our NLCD method based on different models we use for the nonlinear regression. There is kernel ridge regression, support vector regression, or neural network based models, okay? These models are not too complicated compared to the kind of the things that you hear in press about ChatGPT and other. They have billions of parameters. Here you have a simple hidden layer with a few neurons. And so you don't need anything more than that because you only have 3 variables in the system, L, A, and B, right? So these are very simple models. If you use actually complicated models, your performance may deteriorate because you'll be overfitting to your data, okay? So then this green line is a CIT model. So let's just take a look at, and these rows are all for different sample sizes, 300 sample size, 500 sample size, 500 individuals that you're measuring all these 3 variables, and then 1000 individuals, right? So we are able to correctly identify causal relations versus non-causal relations, okay, based on this precision recall curve. So the perfect curve will go like this. It will go on the top and then come down. So you have high precision and high recall, okay. So the more the curve is towards your right top, the better, okay. So you can see here that CIT, for instance, in nonlinear relations, it's usually the worst performing. The area under this curve is less. But then if you have the other 3 versions of our method are better. But if you look at linear relations with large enough sample sizes, all the methods perform similar because CIT was designed for detecting linear causal relations, okay. So this is as expected. I'm going to skip some slides where we do some robustness checks on if you change the random seeds or if you change your data sets, is this trend still observed? And then we do see that the difference between NLCD, which is this pink, and then the CIT, which is this green, are still maintained, okay? So the advantage of NLCD over CIT is still maintained. The more interesting scenario is this unequal variance, where in real world data you may have like a gene that is showing different variance under L equal to 0 and L equal to 1 conditions, okay? So here is a scenario where you can see that this goes from -3 all the way to 1, but this goes from 0 to it actually goes to here to 2, okay? So the one of the L's is more variance than the other one. The LO is a bit shorter, okay? The lesser width. In these conditions, again, CIT was developed under an equal variance assumption. So we are able to, again, because of the way in which we model, we are able to handle unequal variances. And then again, we are able to, in both linear and parabola scenarios where you have different variance, you can see a difference between the baseline and our methods. We are doing better than the baseline methods. And you have equal variance, then CIT, especially under linear relations, will perform better. And then parabola is close to linear in this case, the simulated parabola. So there also both the methods are similar performance, okay? But in unequal variance case, the top row, we are doing better than the baseline methods, okay? And this is the scenario where I said this key conditional independence test, right? So we are able to say that if you fit 2 curves, L equal to 0 and L equal to 1 curves between A and B, right? Using A to predict B and conditioned on L also, right? Using L also as an input, then both the curves are on top of each other, but CIT's prediction shows that you need 2 different linear curves to predict it. And so the conditional independence test passes here but fails here. And that is the main reason why you see the performance difference in this particular plot. In a sense, all of the simulation scenarios, okay, I'll just take the last couple of minutes to just see, say how we apply it to human data, okay. So what we had done is applied it to 10 different tissues with the largest sample size in this data set called GTEx, where they have taken postmortem samples, right? Postmortem different tissues from the same individual and then measured their gene expression profiles. And then looking at the top 10 tissues in terms of highest sample sizes, we then construct trials from these data sets using EQTLs that are identified. EQTLs are simply L that is correlated with A and L that is correlated with B, right? Some slips that are correlated with different genes in these different tissues, okay? And then we look at applying it to either single tissues like the muscle tissue or multiple sets of tissues. Okay. And here is where, like, one of the points I raised before about this has a lot of false negatives, but then whatever we find, we are more confident about that relation. So because of the particular sample sizes, in the particular case of muscle, we were only able to identify 2 relations after applying it to thousands of trials. Okay, so this was a disappointment for us. We went through to all this effort, and then we apply it on the real world data, and then we were able to identify only 2 relations. In East, we had a bit more luck, but I just wanted to show the challenges of these methods, and this is not just for our method, and earlier methods like Finder and CIT also suffers from similar problems. And that is because that actually this table were in the hundreds. But this is one lesson that I wanted to share. The reason I also like to share, I won't say it's a negative result, but more like somewhat like lesser number of priors than usual is if we don't apply a certain filter called a mappability filter. So this is important for anyone doing RNA-seq data analysis, right? So 2 genes in the microarray world, we call it as cross hybridization. The 2 genes which have very similar sequences will get very highly correlated estimates, right? And that is a technical problem, right? And then so we apply mappability filters to make sure that the genes that we put A and B into our trios are not very similar to each other. So once we apply that filter, then a lot of trios get dropped, or a lot of causal relations get dropped. So if we don't apply that filter, like I said, we will get tens to hundreds of relations, but we wanted to be really sure that the relations that we are finding are truly causal relations. And so we find this IRF1 is actually a muscle-related transcription factor. So we find these causal relations. So then we were faced with this challenge of how do we increase the statistical power given current sample sizes, right? So then we looked at regulatory interactions across 10 tissues. So we're trying to pull the signal across all these 10 tissues, so that if a particular causal relation is there not only in one tissue but in multiple tissues, then we share that information and then use that pulled information to make better evidence, to get a better evidence for causal relations, right? Then we were able to get these genes and causal relations between these genes that were concerned across these subsets of tissues. So for instance, this let me take this FAM50A and RPL10P9. So that causal relation FAM50A driving RPL10P9 is supported by whole blood, skin, artery, adipose, and all of these tissues that are listed here. Okay, and then the p-value that comes from the worst of the 4 tests that we have applied, right? So that is kind of called an omnibus p-value, the final causality p-value that we have. That is also shown across these tissues for each of these significantly detected pairs. Okay. And so I'd like to conclude by saying that we have developed a method for nonlinear causal discovery that extends the existing statistical tests that work in a linear setting to the nonlinear setting, right? And applications is promising if you share the information across multiple tissues, okay? With single tissues, there is not enough statistical power, but if you share across multiple tissues, we are able to find these gene-gene relations. Imagine doing this experimentally. If you are trying all possible gene pairs, there is 20,000 in humans, so 20,000 cross 20,000, and then all the 10 tissues, 20,000 cross 20,000 cross 10, with just running it in a computer for a few minutes to maybe 15 minutes, we're able to get all these 10 pairs as hypothesis that could be validated experimentally. Okay, so that is really the power of this method, and then there are other ongoing work that we're trying to do to increase the statistical power further. And to integrate measurement errors along with these nonlinear relations. Okay, with this, I would like to stop and thank you for your attention.
We have time for a couple of questions. So I'll start.
Hi, nice talk, Vani. So did you take into account the reverse causality, number 1? Number 2 is that what was the strength of your instruments? Because most of these SNPs in GTEx are somewhat, it is significant by p-value, but the effect size is very, very small in regulating the expression. So do you think that if you make a threshold for the strength of the instrument, your method will perform better?
Got it, yeah. So those are good points. So when we look at this here, I'm just showing the forward p-value, but we have also tested the reverse p-value, and then make sure that the reverse p-value is greater than a particular cutoff, like 0.05. before we declare. So there's a forward causality p-value for A to B, and then for reverse p-value for B to A, we make sure that only one of them is significant and the other is not significant. So reverse causality is checked and then excluded before we show these results. And then the other point about the strength of the instrument is automatically used in deriving the p-values. So maybe there are other ways to use it. It's a valid point. But currently the methods, the way the strength of the p-values that the method offers already incorporates the strength of the instruments. So if there is a weak instrument, we will get worse p-values. If there is a strong instrument, we'll be able to get better p-values. So maybe one of the reasons also is that we need more sample sizes, could be that these instruments are weak.
Have you tried looking at ChIP-seq or other data sets as both a validation and also to like better figure out parameters framework? Like, because you're just getting two, right? Maybe there's a certain threshold where you have like a training ChIP-seq data that you can use to, figure out more interactions, and that will help you anchor your method.
Yes, so we haven't used ChIP-seq data, but in the yeast setting, in the human setting, we haven't used ChIP-seq data, but in the yeast setting, which results I have not shown here, we have used ChIP-seq and other literature-based methods for identifying protein-DNA interactions, and take a list of transcription factor and all the target genes and other genes, and then we identify whether we are able to pick the target genes, the correct target genes compared to some other random genes which is not driven by the transcription factor. So that we have used ChIP-seq data. The results are promising, but I would say that it is similar to what previous studies have also reported, in the sense that there are more false negatives and less false positives. So this kind of idea of sensitivity being low could not be recovered even with using ChIP-seq data.
My question is hi Mani.
Sorry, yeah.
Yeah. It's quite informative. So my question was very similar to Sriram's. So did you also, is there a possibility, would you attempt having another layer with, say, nucleosome occupancy data or chromatin accessibility, apart from SNPs?
Yes.
So, yeah.
Yeah, I mean, the only way I can see to increase or to address this false negative issue is to use additional data. So ChIP-seq is one thing that we have tried for the yeast setting, but yes, additional data sets could be used. And the way we actually do these analysis, I mean, if we start with only the causal gene networks, we won't be able to do any of the other steps in our research program. So the way we do that is we combine correlation-based networks with literature-based networks based on, let's say, ChIP-seq or other protein DNA or protein-protein interactions, and then have these causal interactions as kind of a cream on the top to say that these are really, really confident edges based on this analysis, and then use the union of those to perform downstream analysis. So this alone will give you certain interesting causal gene pairs, but the field is not at a sufficient point, or the data doesn't have sufficient signal to use only this as a source of your gene networks. So this will give you causal gene networks, but then it has to be combined with other correlation-based gene networks to do kind of empirical analysis.
Yeah, two things. One is interesting talk that thing is that have you been able to get any biological interpretation of this relations that you have seen?
So far it's ongoing. So we haven't. I mean, for some of the interactions like IRF1, and then in a different study where we had an error-aware causal discovery method, we had something called the KLF transcription factors, and those transcription factors were relevant for muscle. So they were known to be transcription factors for muscle. And in each setting, we were able to validate the biology by looking at known transcription factor target relations, where there is much more information on that. And so there also we are able to interpret the biology. But for these interactions, we just recently got it. We haven't delved into the details of support for these interactions.
Yeah. The other thing is what happens when you have, say, A is influencing some D, E, and then that influences C. So not direct relation. But how will that come into this?
Indirect relations are also caught as long as the correlation is strong. So it's not restricted to direct relations alone. Indirect causal relations are also, like multi-step relations are also theoretically can be captured. But if that multiple hops makes the relation weak, then you'll have a challenge capturing it.